Hello and welcome to today's webinar, which is Mischievous membrane proteins and how to tame them. My name is Claire Hattie and I'm an application specialist with NanoTemper Technologies. I wanted to start by first considering why we study membrane proteins. So perhaps I want you to think about what first drew you towards working with membrane proteins. For me, it began as a quest to understand their role in neuroinflammation and neurodegeneration. So I spent most of my PhD doing a lot of rather tedious radioligand binding experiments and trying to express, purify, and reconstitute our target protein into model membranes for structural and functional studies. Now, I'm sure the driver for most of you is very similar, namely to understand the cellular functions of membrane proteins and to contribute towards the design of therapeutics that target membrane proteins. But I know firsthand, as does anyone that's worked with membrane proteins, just how difficult it can be. So, what is the attraction? Well, for one, membrane proteins such as GPCRs, ion channels and transporters, they have a vital role in cellular processes such as signaling and transport, and they make up approximately 30% of the human genome. So for this reason, they're also hugely important therapeutic targets for a diverse range of conditions. And it's estimated that over 50% of current medicinal drugs target membrane proteins. But they are also some of the most challenging targets to work with. Now, some of the key challenges in the structural and functional characterization of membrane proteins and ultimately drug development are their low expression levels, the need to extract and solubilize them from the native lipid bilayer, and the need to maintain their structure and function in a new membrane-like environment, whether this is detergents or liposomes or something like lipid nanodiscs. Now, there are clearly many good reasons to take on the challenge of membrane proteins. So today I wanted to show you some of the ways that we at NanoTemper can help you to overcome some of these challenges in your membrane protein research. By the end of today's webinar, I want to show you how we can help you to characterize membrane protein stability and interactions using small amounts of sample, with some of the key benefits of our technologies being low sample consumption and broad concentration range. I want to show you how you can do fast, label-free screening of membrane protein stability in a range of different detergents, lipids, and other additives with the Prometheus. And this is because Prometheus measurements use intrinsic fluorescence and are buffer independent. And finally, I want to show you how you can study membrane protein interactions in detergents, lipid nanodiscs, and SMALPs in using the Monolith because of some of the key benefits of the Monolith, such as in-solution measurements and measurements in close to native conditions. Now, one of the first challenges in working with a membrane protein is finding a condition in which it's soluble and stable, and ideally one in which it's going to retain its native structure and function. So here the Prometheus NanoDSF is really well suited to measuring membrane protein stability in a range of different detergents or lipids or in the presence of other additives. The Prometheus uses NanoDSF to perform label-free measurements of protein unfolding, and this is using intrinsic tryptophan and tyrosine fluorescence. This means that it's compatible with any buffers or detergents, and it doesn't require the use of any extrinsic dyes. Now, there are also options for simultaneous aggregation detection with back reflection and for particle sizing with DLS. Other features of the Prometheus that make it particularly useful for membrane proteins are the broad concentration range that you can measure and the low sample volumes. So you can measure stability even for very low yield membrane proteins. Some of the other features that make the Prometheus the all-round gold standard in protein stability characterization are the ultra-high resolution measurements and the options for automation. As I've mentioned, the Prometheus NanoDSF, it allows us to measure protein unfolding by monitoring changes in intrinsic tryptophan and tyrosine fluorescence, because the fluorescence intensity and the peak fluorescence of these residues changes due to changes in the local chemical environment. So in this example, if the tryptophan is buried within the protein, when it's folded, but becomes solvent exposed as it unfolds, we see this as a change in its fluorescence intensity and a shift in its peak fluorescence. So that's essentially what we're measuring. Now, the key parameters that we get from a nano-DSF experiment are the melting temperature, or TM, and the onset of melting. And with aggregation, we also get the temperature of onset of aggregation. The first example I want to show you is from a publication where they describe a workflow for high throughput stability screening for detergent solubilized membrane proteins. And this is often a bottleneck in doing any structural studies. In this case, 9 different integral membrane proteins were first purified in DDM as a starting point. They were then diluted in an excess of the 94 other detergent conditions to be screened, and left to equilibrate for 1 hour to allow for detergent exchange. The Prometheus was then used to measure thermal unfolding and aggregation of each protein in the 94 different detergents. So as the Prometheus takes up to 48 samples in one run, this means that each protein could be screened against all 94 detergents within 2 runs. So that would, at normal thermal ramping rates, that would take approximately 3 hours overall to screen each protein against those 94 conditions. The resulting TMs, the onset of melting and the onset of aggregation, they were then used to produce heat maps to show whether each detergent increased or decreased protein stability compared to the DDM starting condition. Here are the unfolding curves for one of the membrane proteins with a selection of the detergents tested. And you can see that there are clear differences in the melting temperature between the different detergents. So in the table here, the arrows indicate whether the melting temperature and onset of aggregation increased or decreased in each detergent relative to DDM. And there are cases where an increase of aggregation onset doesn't necessarily correlate with an increase in melting temperature, such as here, and this highlights the value of looking at both parameters. Now, the authors also correlated the melting temperature with the onset of melting, and this was in order to exclude any conditions where the melting temperature, or the midpoint, was higher, but the onset was much earlier. So in this case, measuring all 3 parameters, that's the TM, the T onset, and the T ag, were really important for optimizing the detergent conditions for these proteins, with the ultimate aim of finding conditions that would likely be more promising for crystallization trials. I also think that this publication provides a really great blueprint for how the Prometheus allows fast, high-throughput screening of detergent conditions, and this is without the need for buffer exchange or for the use of any extrinsic dyes. In addition to screening stability in different detergents, membrane protein stability can also be screened in the lipid cubic phase. Many of you are probably familiar with in mesocrystallization method for membrane proteins. And in this example, this group tested whether it was even possible to measure protein unfolding in the mesophase with nano-DSF. So they measured the TM of 4 different membrane proteins in both the lipid and in detergent and the lipid cubic phase. And you can see here the unfolding curves of one of the membrane proteins tested first in detergent and then in the lipid cubic phase. So you can clearly see that a transition can be measured in the lipid cubic phase. The table here then shows the differences in the TM with the same host lipid, but with different lipid additives as well. So you can see clearly in this case that Prometheus nano-DSF, it allowed for the screening of stability conditions, even in very viscous lipid cubic phase. And this is something that had not been possible with other methods. In order to better understand the critical role of membrane proteins in cellular processes and then to target drugs for them, we often need to understand the interactions of membrane proteins. But as I've mentioned, this can be really difficult because removing them from the membrane can change their conformation and their function. In the following examples, I want to show you some publications that demonstrate how the monolith is well suited to studying the interactions of membrane proteins in solution. This can be even under more challenging conditions, such as with detergents, in lipid nanodiscs, or in SMALBs. I first want to briefly introduce the monolith and microscale thermophoresis in case you're not familiar with the method. So some of the key benefits of the monolith that make it well suited to studying membrane protein interactions are the low sample consumption, the ability to measure a broad range of affinities, so that could be from picomolar to millimolar, the ability to measure KD independent of size or mass of binding partners. So you could measure a small molecule binding to a rather large membrane protein in a detergent micelle or in nanodiscs. The ability to measure in solution and in close to native conditions, and the ability to measure without the need for immobilization of one of the binding partners. Also, the ability to work with any buffer type, so you can work with any buffer that will keep your protein happy. You can also work with any different sample types, so that could be mammalian, that could be yeast and bacterial cultures, fungi, plant extracts, or bioliquids. And finally, some features that make the monolith generally applicable to studying a wide range of interactions. That's that you can measure a broad range of different targets and interactions with the same device. And it's a very easy to use device without any need for ongoing maintenance. With microscale thermophoresis, or MST, the binding events are detected by measuring changes in fluorescence intensity. So we label one of the binding partners and we monitor its change in fluorescence in response to an IR laser-induced temperature change. Now, this change in localized fluorescence, it's due to two factors, which is the thermophoresis of the molecules, or the directed movement within a temperature gradient, and the trick response, or the temperature-related intensity change. And this is the change in fluorescence of the fluorophore in response to a change in temperature. So the overall fluorescence change in response to temperature, this will be different depending on whether the labeled target molecule is bound to a ligand or unbound, because the binding of a ligand to the fluorescently labeled target will change its thermophoresis and may also bind in proximity to the fluorophore and or cause a conformational change of the target. So we measure the change in fluorescence intensity of the labeled target in the presence of various concentrations of an unlabeled ligand, and we plot the relative fluorescence between the steady state before heating and a fixed time point along the curve after heating. Now, by plotting then the relative fluorescence against ligand concentration, we can fit the curve to derive a KD for the interaction. Because MST measurements are done in solution and using small amounts of sample, and it can be done under any buffer conditions, it means that it can be used to study membrane proteins in a range of different membrane mimetic environments. So this could include detergent micelles, bicelles, liposomes, lipid nanodiscs, Saposin lipid nanoparticles, SMALPs, and even in cell lysates. Now I'm going to take you through a series of examples from publications that use some of these different membrane-like systems. And I'm going to begin with focusing on some purified membrane proteins in detergent. Here we have a study of the structural basis of nitrate binding to the 12 transmembrane helix transporter NRT 1.1. Now, this proton-coupled transporter, it belongs to a family of membrane proteins responsible for nitrogen assimilation and nitrate transport in plants, and it switches between a low affinity and high affinity binding states for nitrate in response to falling nitrate levels via a process of phosphorylation. Now, to further understand this mechanism, MST was used in this case to determine the effect of phosphorylation and specific point mutations on nitrate binding. In this case, the transporter was expressed with a GFP fusion and it was purified into DDM. So rather than labeling the protein with a dye, the GFP in this case was used as the reporter fluorophore for MST experiments. What we can see in the red curve is the binding of the wild-type transporter to the nitrate with a KD of approximately 1 millimolar. We can then see with 1 point mutation that this doesn't affect the binding, so we have a KD again of around 1 millimolar. But then we can see that with a mutation of the histidine 356 to alanine, that it abolishes the binding altogether. So by combining MST and crystallography, they revealed the key amino acid residues for nitrate binding and subsequent transport. So this helped to further unravel how the phosphorylation of the transporter allows it to switch between binding states. This example also nicely demonstrates how sensitive MST is because it allowed the measurement of a very small ligand binding to a large target in solution, even with a weak affinity. The next example uses both NanoDSF and MST to help understand the structural basis of drug and peptide binding and transport. Now, the human PepT1 transporter is an important pharmacological target due to its role in uptake and transport. So in this study, researchers used a bacterial homologue, the DTPA, in order to better understand drug binding and transport of the human protein. First, they used the Prometheus NanoDSF to screen for nanobodies that would increase the stability of DTPA in order to aid in crystallization and structural determination. They then performed a thermal shift assay with 2 prodrugs, and you can see in the graph here that both prodrugs increase the stability of the DTPA, resulting in a shift of either 3.8 degrees or 6.2 degrees. They then also performed a larger thermal shift assay with the DTPA in the presence of a series of tripeptides, dipeptides, and drugs. And you can see that the Tm of the protein changes in the presence of many of these potential ligands. It also reveals that overall, the tripeptides were the most stabilizing. The implication here is that those peptides or the drugs that cause a thermal shift are doing so by binding to the DTPA. So these interactions were then characterized further with MST. Label-free MST was used in order to quantify the binding of a variety of the peptides and drugs to the DTPA and to determine the residues involved in binding. So in this case, the MST was carried out on purified DTPA in DDM, and rather than attach a fluorophore, the intrinsic fluorescence of the protein was used with label-free MST. So here we have the binding curve of the DTPA binding to one of the tripeptides, and that's the DTPA either by itself or in complex with the stabilizing nanobody. And you can see that in both cases, there's a KD of around 58, 59 micromolar for the interaction. So by combining MST with various mutants and the crystal structure, they revealed the binding site on DTPA for one of the prodrugs. And this helped then to provide additional insights into the drug binding of the human PepT1 in order to aid in drug design. As I've mentioned, one of the biggest challenges in studying membrane proteins is how removing it from its native lipid environment can fundamentally change its structure and therefore change its function or affect its function. Now, there have been significant advances in developing systems that allow us to study membrane proteins in increasingly near-native membrane environments, and this could be lipid nanodiscs, sappers and lipid nanoparticles, or SMALPs. So in the final part of our session today, I'd like to show you a few more examples, recent publications, of how MST can be used to study binding interactions in these membrane mimetics. The first example demonstrates how MST can be used to measure membrane protein interactions in sappers and lipid nanoparticles. In this case, the membrane proteins in question were reconstituted into sappers and lipid nanoparticles by first mixing detergent solubilized lipid, detergent solubilized membrane protein, and then the lipid binding protein sapacin A, which forms disc-like particles in the presence of lipids. The detergent is then removed, and this forms a lipid nanoparticle around the membrane protein that's encircled by the sapacin A. So they first did NanoDSF, and this revealed that the membrane proteins were more stable in the sapacin lipid nanoparticles than in detergent, as you can see here. And again, label-free MST was used to show that the sapacin lipid nanoparticle reconstituted membrane proteins retain their ligand binding capability. And in this example of the PepT protein in either DDM or in sapacin lipid nanoparticles of different lipid compositions, we see it binding to a dipeptide, so it retains its binding capability. So again, we can see that both NanoDSF and MST are well suited to studying stability and interactions of membrane proteins, and that could be either in detergent or reconstituted into sappers and lipid nanoparticles, without the need for immobilization or the need to use dyes. GPCRs are obviously a very important target class for drug discovery, and here we have an example of how lipid membrane fluidity affects the ligand binding activity of the human serotonin receptor. In this case, the serotonin receptor, it was purified into detergent and then reconstituted into nanodiscs of different lipid compositions. Finally, the protein lipid nanodiscs were labeled with NanoTemper's TRIZ NTA dye, which specifically labels the his tag on a protein. They then used MST to measure the binding between serotonin and the serotonin receptor, reconstituted into different nanodiscs of different lipid compositions. And this revealed that the binding is different depending on the lipid composition of the nanodiscs. And indeed, you can see that there is no binding at all detected in the DMPC nanodiscs. And what this indicated was that the decreased membrane fluidity in DMPC nanodiscs, it may prevent the conformational change of the receptor into the active form. So this shows that membrane environment is really critical when characterizing GPCRs in vitro. And in this case, MST allowed the GPCR lipid nanodiscs to be studied in solution, which meant that they could retain their ability to change conformation between the inactive and the active form. The final two examples I'd like to show you how MST is now being used to study membrane proteins in SMALPs. So SMALPs are a membrane mimetic that retain some of the native lipids surrounding a membrane protein. The SMA polymer is used almost like a cookie cutter that will form discs around membrane proteins containing some of their surrounding membrane lipids. You can then use size exclusion chromatography and affinity chromatography to isolate just the membrane protein of interest within the SMALPs, allowing for detergent-free purification of membrane proteins. And in this paper, this method was used to extract and purify a very low expressing human dopamine receptor into lipodiscs without the use of detergent. They then used MST to measure the binding of the neurotensin peptide to the dopamine receptor in lipodiscs. And in this case, the neurotensin peptide was labeled with the Cy5 fluorophore and used as the target, while the dopamine receptor was titrated. Now, while the interaction didn't reach full saturation, it did show for the first time that there was a specific interaction between neurotensin and the dopamine receptor in vitro, with a KD of just over 300 nanomolar. It also really nicely highlights how MST can be used to study the interactions of even very low expressing GPCRs in a more natural membrane environment, and due to the low sample consumption requirements and the ability to measure in solution under any buffer conditions. Finally, we have an example of how MST has been used to study small molecule binding to a ligand-gated ion channel in SMALPs. In this case, it was the human glycine receptor, and it was expressed with a his tag, and detergent-free purification was carried out in SMALPs. So for MST experiments, they used NanoTemper's TRIZ NTA dye to label the glycine receptor via its his tag, and then they measured the binding to glycine and taurine. So here you can see the binding curve for the glycine receptor binding to glycine, with a KD on EC50 of around 65 micromolar. So again, this nicely highlights how MST can help you to study the ligand binding interactions of different classes of membrane proteins in SMALPs, even when binding to small molecules. And again, here the use of the TRIZ NTA dye, it allows for a gentle site-specific labeling of the target membrane protein within the SMALP. I hope that you've now had a good overview of how MST, and for most of these systems, NanoDSF, allows you to more easily work with membrane proteins in a range of different membrane-like environments. To summarize, how can we help you finally tame your mischievous membrane proteins? Today, we've seen how to overcome some of the key challenges in working with membrane proteins, namely their low expression levels, the need to extract and solubilize from the native lipid bilayer, and the need to maintain structure and function in a new membrane-like environment. So we can help you by giving you the tools to characterize membrane protein stability and interaction using small amounts of sample, to perform fast label-free screening of membrane protein stability in a range of different detergents, lipids, or other additives with the Prometheus, and to study membrane protein interactions in detergents, lipid nanodiscs, and SMALPs with the Monolith. With that, we conclude our exploration of mischievous membrane proteins and how to tame them. You can find more membrane protein resources on our website by looking for our membrane protein spotlight, and you can also learn more about our full range of products and applications. Please feel free to also reach out to me or your local sales and application specialist via the website. Thank you for your attention today, and good luck with your mischievous membrane proteins.